Hi there! Welcome to Utah Valley University's Herbarium, located in the Science Building, SB 277. My name is Hannah Veldkamp, and I've been working here as a student curator for about 2 years. And how I got into it is I'm a botany major, so I was interested in plants, and I knew that herbarium worked with plants. But other than that, I didn't know too much about it. In my 2 years working here at the herbarium, I've been able to learn more about what we do here and how it works. And you could have the possibility to work in here too. You could volunteer, work, study, do research. And I'll be able to explain those things to you when we go through the tour of the herbarium, and you can learn more about yourself by coming in. In this office back here, we've got our herbarium director curator, and it's Erin Riggs, and she is amazing. What do you do as director curator of the herbarium?
Well, as director curator of the herbarium, I'm in charge of, the entire herbarium, the decisions regarding our collections, what we need in our collections, curation techniques, trying to keep up with the best conservation techniques for our museum, as well as hiring and training students as student curators in our herbarium here on campus.
How did you get interested in herbaria curation?
Well, I initially was very interested in botany, of course. I was going to university and I was taking all these different classes trying to figure out what I wanted to do, and I fell in love with botany, and that led me to systematics, evolutionary studies. That lends itself to herbarium work because museums and herbaria are all about the relationships of among and between species. And I worked in several herbaria, Oregon State University, Hoyt Arboretum Herbarium, and I got some excellent training in herbarium curation, and also museum training is really important in regards to getting into herbarium curation.
Professionally, how does one become a director curator of an herbarium?
Well, you need to get a degree in, usually it's an evolutionary biology degree of some kind. Mine is an evolutionary botany degree in plant systematics. And also, as I mentioned before, how I got into it was getting training and working in the herbarium, different herbaria, and museum curation training, which I think is actually very important. Most curators and director curators have PhDs. I have a master's degree. Due to my combination of training and experience and teaching at open enrollment, lent itself to me getting hired at UVU as the director curator.
The amazing Erin Riggs, folks.
Anna, you're too much.
I know. First question we should answer, what is an herbarium? An herbarium's like a plant museum, plant library, plant morgue, because the plants are dead, like this one here. It's where plants are collected from wild or cultivated populations and brought into the herbarium, curated, and stored in a stable environment so we're able to further study them. So the first step in selecting a specimen to be a part of our herbarium is first going out into the field and finding a specimen that best represents that population. And when we find the plant that we want to collect, we try to get information that is listed on this data sheet. We like to know who collected it, when it was collected, where it was found, associated species, just everything and anything on the plant, so we have a knowledge about the plant from the field. And when we dig up the plants, we try to get roots, shoots, flowers, and fruits, because we want all the information as much as we can. And we know it's not possible to dig up a tree for the herbarium, so we try to cut off a sample that will best represent this plant. And once we've selected our plant and we've cut it off or dug it up, we put it into a plant press like this. It starts with a wooden frame here, and then we'll put in a piece of cardboard, and then the specimen itself will go into a newspaper. And then on top of the newspaper and the specimen will be a piece of felt, foam, and another piece of cardboard. And that will be repeated for other species to follow. And after that, we put on the second wooden frame, and we've got our straps, and we have someone stand on top of it to get the specimen as flat as possible. The reason we have someone stand on top of the press is to prevent morphological distortion when they're drying. And what we're preventing is the plant organs, the flowers, the fruits, and the leaves and the stem, just all of it, from getting wrinkled and shriveled. Because if it's wrinkled and shriveled and we need to go in to identify the plant, it's difficult to access the organs, especially in the flowers, and key identifiers when we're IDing the plant. If we can't access those organs, it's pretty difficult to narrow it down to what plant it is. And after it's put in here, it's brought into the herbarium and placed into the dryer for a couple of days to a couple of months, depending on how wet the specimen is. Once the plant is done drying, we'll open up the plant press, and it's like Christmas because we get to rediscover our plants again. And we'll take the plants out and we'll begin to ID them using different flora. And each of the floras, there's a dichotomous key, which helps to break the code of the plant so we can better identify what the plant is. And so what we'll do is we'll use a key, and I'll give us a couplet that has two choices. And in each choice, we get to pick which one better describes our plant. And once we find the one that best describes our plant, we work our way through the key until we find out what our plant is. And the reason we use multiple flora is so we can confirm that our identification is agreed in multiple books. And after it's identified, it's getting ready for mounting time. Mount, 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 mount. We have two examples of specimens that have already been mounted. But what we'll do when we get ready to mount them is we'll lay the specimen down on archival paper. And the reason for archival paper is we don't want anything to perturb the specimen later on or off gas, which could harm the plant specimen. We want to preserve it as long as possible. So we'll put our plant specimen on, and what we currently do is we use a linen tape instead of glues that were used in the past, because linen tape doesn't harm the specimen as much as glues are doing. And once we've got the specimen down itself, we'll put on the label with the information we collected out in the field and the name that we found with the floras. Then we'll give it an accession number, give it our stamp for our herbarium. And then the fragment folder, another new addition, we use that to store any broken specimens that may have fallen off. So we can use it later in DNA barcoding and destructive sampling, but also so we can keep all of the specimen together. Our lovely origami. And then it gets ready to be put in the freezer for about a week. Now I know that sounds a little strange to put a specimen into the freezer, but the reason we do that is to kill off any fungal, bacteria, or insects that may have survived the drying process, because we don't want them to infect the rest of our specimen. And after the freezing process, it's ready to file. So after the plants are done in the freezer, we file them away in the cabinets. And we first organize them into non-vascular, gymnosperm, and angiosperm. Non-vasculars are plants that don't have vascular tissue, so they don't have xylem or phloem to help transport food and water to the different parts of the plants. An example of that would be lichens and mosses. And this is our lichen, example of a moss, and it's such a pretty baby from New Jersey, and you can see all the cute little lichens. It's even got a secret little moss on it, because when you take the sample, it's not going to be perfect because you actually take the substrate. So I'm pretty sure this is on bark. So yeah. And then our other group I mentioned was gymnosperms. And you break down the word gymnosperm and it means naked seed. So those are seeds that don't have ovaries covering them or a fleshy fruit covering the seed. So an example of that would be your conifers and pines, like this plant here. And then the other group we divide it into is angiosperms. And you break that word down and it's coated seed. And those are the flowering plants and they are enclosed in carpel. An example of our angiosperms are any flowering plants that you see. And then after it's organized into those three groups, they get organized alphabetically by family, genus, and species. So you really need to know your alphabet if you're going to start filing in here. So why is all of this important? Why is the herbarium important? Why are vouchers important? Why go through all of this work? What it comes down to is the information we can get from the plants themselves and the easy access to have plants here on campus that you wouldn't normally find close to campus. When it comes to the individual vouchers themselves, it provides a biological historic record that can be kept in perpetuity. It even supports people's research. If a scientist has doubts on a plant that someone was researching, they can go back to the herbaria that had that plant, look at that voucher, see if the plant was ID'd correctly, and if the information on it was correct. A voucher withstands the peer review process, so it's a great support in your research. And there's a lot of information we've been finding out with herbarias that we didn't know when they were originally created. For example, we've been learning more about climate change. We've been finding out that plant size has been decreasing, and the phenology time of plants, the flowering time, has been changing, and the amount of carbon we find in the plants is increasing. And as for herbaria themselves and the future they have, there could be importances to the herbaria we have not found yet. And what's awesome is an herbaria is here to engage anyone's curiosity to find out their own possibilities of what they want to do in research. So the possibilities are endless and up to you to pick. Some of the faces you'll see in the herbarium is Director Curator Erin Riggs, and she's studying floristics and making a comprehensive list of the Grand Gulch Bears Ears area. Dr. Ashley Egan, a field botany professor doing biodiversity research. Dr. Michael Rotter, a field botanist and plant anatomist doing mimulus research. Dr. Elma Laney, a plant virologist. Dr. Michael Stevens, plant ecologist and director of the Capitol Reef Field Station. Dr. Olga Kopp, plant physiologist and ethnobotanist, doing research of fungal and bacterial biofilms, in addition to plant tissue cultures of endemic endangered species. Dr. Jeffrey Zahn, mycologist and microbioecologist, studying plant and fungus interactions. Josh Schneider, botany lab manager and native garden research manager. Sam Doty, greenhouse manager. We have two professor emeriti, Dr. Robert Robbins and Dr. Jim Harris, a founder of the UVU Herbarium. When it comes to research in the herbarium, we've got a couple different opportunities that you can join or even come up with your own. We have DNA barcoding, which is the process of identifying specimens based on their nucleotides and the diversity shared in the DNA sequences. It's like taking a fingerprint of the DNA. We also have conservation and museum work. We have a student that's been looking at glues and how they affect herbarium specimen and seeing which glue or even linen tape is going to be better to preserve the specimen itself. We have a partnership with Loveland Living Planet Aquarium. We're even doing a project where we're looking at their tropical plants and IDing them, and also taking samples and making vouchers of them to store in the herbarium so we can have a record of what plants that they're using there. We even are researching phenology, which is looking at the flowering times, when the leaves come out, and when the leaves fall off on plants. And how we do that is we look at current living specimen and compare them to voucher specimens that we have here in the herbarium, or even other herbaria via the interwebs. We even do index semina, which is a special seed collection from our native garden, and we trade with other native gardens that have index semina, and we're trading really neat seeds for more cool seeds so we can even increase the diversity we have in our garden. We even are doing the sketchbook project through the Brooklyn Art Library. And the Brooklyn Art Library's goal for their sketchbook project is to help encourage artists to show their creativity. And our goal for our herbarium with the sketchbook project is to show the intersection of art and science. And of course, we're always open for new ideas of research or other ideas you think we can do here in the herbarium. When it comes to student resources we have here in the herbarium or in the same building, we have our reference library, which is about 1,000 books large. We also have the greenhouse located on the 3rd floor of the science building. We have vouchers and specimens that you're welcome to reference, and if you want to look at some of the specimen, we have student curators and the director curator that can help you browse our collection. We also have professors, staff, and student curators that can help you with questions that you may have about the herbarium. An additional resource specifically for botany majors, we are honored to have the Kimball T. Harper Scholarship. It's a scholarship that assists with a year's tuition, and applications are due February 1st of every year. So I suggest you apply if you're interested in the scholarship, and especially botany majors, because this one is for you. And if you have any questions about the scholarship, contact Dr. Michael Stevens. We have volunteer opportunities for any major. We have botany and biology, photography, psychology, education, many different art majors. So any major, we'll take it. And after you're trained to volunteer in the herbarium, you can help with filing, which is learning the Latin names of plants and organizing them alphabetically in the cabinets, which I think is a fun task and I quite enjoy because I get to see new plants, see where they're located, and even look at some of the folders and see what's inside them. You can also help with mounting, which is using your creative side and putting plants on a voucher to give them their permanent home. You can also help with keying out plants, which helps with decision making and visual literacy. You can also, while going through all of these processes, become more familiar with plant families and be able to identify like what plants go to what family. And most importantly, you become a part of the botany nerd family, which I think is important because we're a cool group of people. And what's awesome about our volunteer opportunities is you can cater it to what best fits you and what you're interested in. So we'd love to see you in here and volunteering. In addition to volunteer opportunities here in the herbarium, we have work study positions here. A position that I have is assistant curator. And my focus and what I've done with the position is I focus on outreach. So I help give herbarium tours. I create outreach displays that we have here in the herbarium. I've helped with outreach programs. For example, one of those is creating a bingo that an age group of kids can participate in while they're exploring the herbarium so they can learn about the herbarium and also enjoy what they're doing. I also help with filing and mounting and keying plants. And I think one of my favorite opportunities is learning about fellow students' research here in the herbarium, so I've been able to see what they're doing in their research and add that to my knowledge bank of what I tell people when I'm doing outreach. And back here we have our amazing assistant curator that specializes in photography. She takes pictures of our specimen back here to make sure we've got a good record of our database online. And it's the amazing Alyssa, who is not right here because she's been behind the camera.
It's me!
Mario!
Mario!
Tell us a little bit about yourself.
So my name is Alyssa Chamberlain, and I'm the assistant curator specializing in photography back here.
What do you do in the back of the herbarium?
That is a good question. So, I do anything that relates to photo or video, like this, like this video. I also take photos of the herbarium specimens in this light box here. So we upload it to the database where people can go and look at the specimens online with all the info and then a high-res photo of it. And like other herbarium can look at it and get info from it and stuff like that. So it's a really important process because especially nowadays where everything is digitized, it's really good to have everything online and saved, not just to have it as a backup, but so that anyone can look at it. It's a way to like share it with the world and get a lot more research out of it. So very important.
What's been your favorite part about working in the herbarium?
I mean, I'm a photographer, so I love taking photos, so anything that revolves around that is great. But I do like using this guy, the light box, because I've been able to take some specimens from here in the herbarium as well as the greenhouse that we have. I can take pictures of the live stuff and have that as well for people to reference. I did a whole project where I took like really close-up photos of these specimens that were like fairly small usually, and I had to take like 40 plus photos just to get like the depth of field, because I was so close that it only showed a small depth of field for each photo. So I took a bunch of photos and then I image stacked them in Photoshop to get a full depth of field range. And then I printed them out super big so you could see all the detail. That was a cool project that I did here. So that was fun. So we also have this really cool microscope camera, the Olympus SZX10. So this is a microscope, and then we have this camera on top of it. And so we can put it on this program, see exactly what we're getting a photo of. Like with a microscope, you're only getting a part of it in focus. So with this technology, we can take, multiple photos of multiple parts of it in focus, and then like image stack them so that it's all in focus. So you get this nice clean photo with lots of detail. So we've been able to use this for a lot of different things. It's been super handy. I love this thing.
And this has been the amazing Alyssa Chamberlain, assistant curator specializing in photography. Thank you for letting me share my knowledge with you about the herbarium. I'm glad you guys could stop in and see what we're about, and we hope to see you in here soon. Is that pollen or the glues? So under here it's got like yellow. Oh, that's definitely pollen. Those are the male cones and these are the female cones. My brother on our camping trip, how he was so excited to know the difference. He's like, party trick. Woo!
Of these specimens, really tiny specimens close up because it's a microscope, because that makes sense, because that's what they do. Anyway.
And I am not tall enough to reach it without causing damage. This more closely, and as you can see, I'm pretty sure we've got some pollen grains here, which are pretty interesting, very interesting. Lovely pollen grains, and then you see the ventation of the petals, and oof, it's so pretty. Like, I'm in heaven, man. You've got Hannah, who's pretending to be the photographer now. Well, filmonography. There's a reason you're the photography majors and I'm the botany major. All the prettiness, I just want to love it. This is so cool.